Hi everyone, today we are going to study about the regulation of blood pressure in human beings and the various mechanisms that play in the body. So, starting with the process, let us understand the definition of blood pressure. It is the lateral force exerted by the circulating column of blood on the wall of blood vessels, and it is the product of cardiac output and the total peripheral resistance in the body. And cardiac output itself is the product of heart rate and stroke volume. The blood pressure in our body is under the regulation of three major arms. ये तीन मेजर आर्म्स के कंट्रोल में होता है जो डिफरेंट टाइम स्पैन पे ऑपरेट करते हैं. देर इज़ अ शॉर्ट टर्म रेगुलेशन ऑफ ब्लड प्रेशर जो सेकेंड्स टू मिनट में ऑपरेट करता है. देन देर इज़ एन इंटरमीडिएट टर्म रेगुलेशन ऑफ ब्लड प्रेशर जो मिनट्स टू आवर्स में ऑपरेट करता है. और फिर देन देर इज़ अ लॉन्ग टर्म रेगुलेशन ऑफ ब्लड प्रेशर जो आवर्स टू वीक्स में ऑपरेट करता है. ये तीनों आर्म्स आपस में कोरिलेटेड और स्ट्रांगली इंटरट्वाइंड होते हैं. जैसे इंटरमीडिएट टर्म रेगुलेशन जो होता है इट इज़ मीडिएटेड बाय किडनी एंड द क्रॉनिक आर्म ऑफ दिस रेगुलेशन ओनली बिकम्स अ लॉन्ग टर्म रेगुलेशन इटसेल्फ. सो लेट अस rapidly look at what are the different mechanisms of these regulation. The short term regulation is mediated by the baroreceptor reflex. It is also mediated by chemoreceptor reflex and the CNS ischemic response, which is also called as the last ditch response of the short term regulation, and it is the strongest short term regulation of blood pressure. Then we have the intermediate term regulation. It is mediated principally by the kidneys through renin, angiotensin, aldosterone system and the ADH that is secreted from the posterior pituitary. But at this point of time, the principal action that we are looking at is a vasoconstrictory effect of both angiotensin 2 and the vasopressin that is ADH. The long-term regulation is nothing but extended function of the renin, angiotensin, aldosterone system that acts by sodium and water excretion and maintenance of sodium balance and by maintenance of ECF volume. Additionally, we have other mechanisms like capillary fluid shift and stress relaxation response which take part in intermediate regulation, and ANP, BNP which are atrial natriuretic peptide and brain natriuretic peptide which take part in long-term regulation of blood pressure. So now you have understood the summary of the blood pressure regulation. This image on your screen is basically a summarized answer of BP regulation. But let us learn a little bit about it in detail. So BP regulation in humans is principally in control of autonomic nervous system. And in autonomic nervous system, the center of BP regulation, it is called as the vasomotor center. वेजोमोटर सेंटर हमारी जो ब्रेन स्टेम होती है उसके रेटिकुलर एक्टिवेटिंग सिस्टम के अंदर प्रेजेंट होता है और ये एक्सटेंड करता है मेडुला में और लोअर 1/3 ऑफ पॉन्स में. ये डिवाइडेड होता है 3 मेजर एरियाज़ में. पहला कहलाता है द प्रेशर एरिया और द वेजो प्रेशर एरिया विच इज़ प्रेजेंट इन द रॉस्ट्रल वेंट्रल मेडुला और RVLM. दूसरा होता है डिप्रेसर एरिया ऑल्सो कॉल्ड एज द वेजोडाइलेटरी एरिया विच इज़ प्रेजेंट इन कॉर्डल वेंट्रोलेटरल मेडुला और CVLM. ये दोनों आउटपुट रीजन्स होते हैं. और हमारा फिर एक सेंसरी रीजन भी होता है वेजोमोटर एरिया का दैट इज़ कॉल्ड एज न्यूक्लियस ट्रैक्टस सॉलिटेरियस बिकॉज न्यूक्लियस ट्रैक्टस सॉलिटेरियस इज़ द न्यूक्लियस which receives the inputs from the peripheral receptors like baroreceptors and chemoreceptors to maintain BP all over the body. We also have an additional center that is cardiac inhibitory center or CIC that augments the heart rate in response to changes in BP. So this was about the regulation of BP by the autonomic nervous system. Other than that, there are certain cortical areas which also influence BP. For example, hypothalamus, which is called as the master of the master gland, it can both increase and decrease BP via its projections to the medulla. Prefrontal cortex, since it is a seat of emotion, since it is a seat of calculative thinking in states of stress, anxiety, it can again affect BP by increasing and decreasing it. Then limbic system, which is called as a seat of emotions, it can also increase and decrease BP in response to various emotions. The most important cortical part that causes an increase in BP is the motor cortex. So motor cortex causes an anticipatory increase in BP before even initiation of movement, and this is called as the alarm reaction of the body. It prepares the body before an anticipatory action has taken place. So this was a summary of the cortical areas of the brain which are other than autonomic nervous system and are responsible for regulation of BP. Now we can come on to the principal areas and the principal mechanism of regulation of BP. Let us start with short-term regulation of BP, which takes place within seconds to minutes after the BP fluctuation takes place. The first mechanism that we are going to study here is the baroreceptor reflex. The word baro actually stands for pressure itself. So these receptors, they detect the change in pressures. So now the question arises, where are baroreceptors located? Baroreceptors are located all over the large vessels of thorax and neck. So all the large vessels like carotid arteries, subclavian artery, and descending thoracic artery, descending thoracic aorta, arch of aorta, they all have baroreceptors located in them. But these baroreceptors, they become maximally concentrated in two important positions of our body. One of them is called as the carotid sinus. The carotid sinus is present at the bifurcation of common carotid, where it divides from internal and external carotid artery. This is the carotid sinus. And the other side is the aortic arch. In the aortic arch, it also has baroreceptors. These two sites in our body are the principal baroreceptor zones which signal to the vasomotor area. And these sites also have other type of receptors which are called as chemoreceptors, which we are again going to study, which also help in, along with the baroreceptor reflex, in the management of BP itself. So baroreceptors, these are example of stretch receptors. So these are nothing but stretch receptors. And what are the type of receptors these are? These are called as spray type receptors. And hence they belong to a family of mechanoreceptors. What happens actually? So whenever there is a change in arterial pressure, usually these baroreceptors continuously fire at a rate of about 0.2 to 1 hertz. 1 hertz. That is, this is the rate of firing of these receptors continuously, and this is called as the normal vasomotor activity. And this normal vasomotor activity produces what we call as the vasomotor tone of our body. What happens in situations when arterial pressure decreases? So for example, when somebody suddenly stands up, there is a pooling of blood in the legs, and there is sudden reduction in arterial pressure in the thorax and the neck, and there is reduction of arterial pressure at these receptors. This reduction in arterial pressure, it causes a reduction of baroreceptor firing. The reduction in baroreceptor firing, it is then transferred via certain nerves to the medulla, to the vasomotor center. In the vasomotor center, there is increase in sympathetic activity and there is decrease of vagal activity. So what it causes? Decrease in vagal activity increases the cardiac output by increasing the heart rate and increasing the conductivity in heart, while increase in sympathetic activity increases the vascular resistance all over the body, and this increases the BP and it counteracts the fall in BP that occurred due to standing. Simultaneously, when there is an increase in BP, there is increased baroreceptor firing, and increase in baroreceptor firing does the opposite job. It decreases sympathetic activity and it increases the vagal activity. So the nerve that carries these impulses from the baroreceptor, the nerve that carries from the carotid sinus, is called as Herring's nerve, and Herring's nerve is a branch of glossopharyngeal nerve, that is cranial nerve 9, and the baroreceptor nerve that carries it from the aortic baroreceptor, that is called as Sion's nerve, and it is a branch of vagus nerve. both of the nerves, they relay their messages into the vasomotor center, and where it is processed and the specific action is taken. Now these baroreceptor reflexes are quite important. Their principal function in our body is not for the long-term regulation of BP, but actually to prevent minute small fluctuations in our body. This image beautifully explains that. So this is the BP reading of a dog. So dog is an experimental animal in which this is the BP reading of a dog, and you can see in a normal dog the BP remains more or less normal, it is not much fluctuating. But when in a dog whose baroreceptors are denervated, so they become non-functional, the BP becomes so erratic and it becomes non-linear, as well as it becomes highly erratic. So the body is unable to cope up with the minute fluctuations of BP when baroreceptors do not work, and hence the principal function of baroreceptors is to cope up with minute fluctuations of blood pressure. Simultaneously, it is also important for you to know that these baroreceptors, they do not continuously since they are there for the functioning for minute fluctuations of BP, they do not work for chronic changes in BP. So for example, if there is a change in BP that exists for more than few hours to few days, their firing rate returns to normal. That is, they adapt, and they adapt within 1 to 2 days, and hence they do not become part of the long-term BP regulation of our body. But there is one more arm of baroreceptors that actually modulates the renal activity, and this long-term modulation of renal activity by baroreceptors, it can actually alter the mean arterial pressure in the long term. That is again indicated by this graph. This is a graph which shows the normal BP of a normal dog with intact baroreceptors. The second graph, that is a right line, shows the BP in a denervated dog who do not has baroreceptors. So you can see that BP in a normal dog, it follows a narrow range, while that in case of a dog whose baroreceptors have been denervated, it has a wide range. This shows that how baroreceptors are responsible for maintaining and preventing minute fluctuations of BP. So this was all about baroreceptors. Now we also came to know about other receptors, that is the chemoreceptors, that also exist in the same regions. Alongside baroreceptors, we also have certain bodies which are called as carotid bodies. Carotid bodies or chemoreceptors. Similarly, we also have aortic chemoreceptor bodies which are present in the aortic arch, which are about 1 to 3 in number, and these are sensitive to pH and concentration of CO2 and concentration of O2. Principally, they are sensitive to the concentration of oxygen and concentration of hydrogen ions, that is indicated by pH. So what actually happens, these also function in a similar manner, but the range or the BP level at which they activate is different. So let us see. So for baroreceptors, typically the carotid baroreceptors, they activate after a pressure that is around 60 mm Hg to 180 mm Hg. They can only operate in this pressure range. Then we have aortic arch baroreceptors. Aortic arch baroreceptors, they only activate in a range that is 80 mm Hg to 180 mm of Hg. While the chemoreceptors that we see, they activate below a pressure of 90 mm of Hg. So you see, since carotid baroreceptors and aortic arch body baroreceptors, they can only function in a narrow pressure of 60 to 180 and 80 to 180 range, Below these pressure, whenever the pressure of our body falls below this pressure, only then the chemoreceptors of our body help in the regulation of BP. So this involves the baroreceptor and chemoreceptor and how these reflexes help in the regulation of BP. The next mechanism that we are going to look up is the CNS ischemic response. So it is triggered when the cerebral blood flow falls below 60 mm of Hg. Basically, it is such that that cerebral blood pressure, when it falls below 60 mm of Hg, there is marked reduction in cerebral blood flow. This causes accumulation of lactic acid, accumulation of CO2, and accumulation of hydrogen ions. And this leads to massive sympathetic activation, and it can cause the BP to rise to 250 mm of Hg. And an example of CNS ischemic response is Cushing's reflex. So Cushing reflex, it is due to nothing but increased intracranial pressure. Increased intracranial pressure is responsible for a reduction of cerebral blood flow, and that causes bradycardia, hypertension, and irregular respiration. Let's look at how this mechanism happens. So an increase in ICP, it causes reduced blood flow. This reduced blood flow, it initiates the vasoconstrictory effect of CNS ischemic response. And why it happened? It happened because there was decreased blood flow to the brain, there was decreased oxygen to the brain, decreased pH, and increased PaCO2. There is sympathetic activation. Sympathetic activation caused hypertension, and the increased PaCO2 and decreased pH, and decreased oxygen, principally decreased oxygen, was responsible for respiratory suppression, and this caused irregular respirations. Because of sympathetic activation, there was widening of pulse pressure, which caused an increased stretch on the baroreceptors, and these baroreceptors caused parasympathetic activation, which caused bradycardia. And therefore you can see the simultaneous activation of sympathetic and parasympathetic system due to increased ICP is the cause of hypertension along with bradycardia in Cushing's reflex. Okay, so let us move on to the other reflexes. So other than that, there are some additional reflexes which are called as the low pressure reflexes of heart. The low pressure reflexes, which are mediated by the low pressure receptors. These are present in the walls of atria and pulmonary vessels, and they also function in a similar manner to baroreceptor reflex, but the measurement of these reflexes is quite difficult and it is not usually done. There are additional reflexes which actually also help in short-term regulation of BP, and one of them is Bijold-Zarisch reflex. So Bijold-Zarisch reflex, it causes three important things. There is bradycardia or reduction in heart rate, there is hypotension or fall in BP, and there is peripheral vasodilation. This is the triad of Bijold-Zarisch reflex, and let us look at what causes it to happen. It can be caused by either activation of mechanoreceptors or chemoreceptors. So mechanoreceptors or chemoreceptors. Mechanoreceptors can be activated by conditions which actually cause a decrease in preload. Just a minute. So mechanoreceptors can be triggered by anything that is causing decreased preload. Mechanoreceptors can be triggered by anything that is causing decreased preload or increased sympathetic activity, which causes vigorous ventricular contraction, or it can be activation of chemoreceptors that could be due to release of bradykinin, reactive oxygen species, prostaglandins due to myocardial ischemia. All of this causes activation of vagal afferent nerve, and this vagal afferent nerve causes bradycardia, hypotension, and peripheral vasodilation, and this is Bisol-Zaris reflex. There is one more reflex which activates in an antagonistic manner to the baroreceptor reflex, and that is called as the Bainbridge reflex. Bainbridge reflex is basically the acceleration of heart rate by increased right atrial pressure. So basically, when there is intravenous infusion or there is sudden intravenous infusion causing an increase in circulating volume, this causes increase of right atrial pressure, and right atrial pressure, this causes increase in heart rate, and this increase in heart rate is called as the Bainbridge reflex. You see that increase of cardiac output due to fall of intravascular volume was causing baroreceptor reflex that was associated with fall of heart rate. So Bainbridge reflex and baroreceptor reflexes both are examples of antagonistic mechanisms. Both are actually associated with due to alterations in volume, where Bainbridge reflex is activated by increased circulatory volume. Baroreceptor reflex is due to decreased volume that is present around the baroreceptors. So this summarizes the short-term regulation of BP. Coming to the intermediate and long-term regulation of BP, we have already seen different mechanisms. So let us club the two processes and study them together. These are principally maintained by the renin-angiotensin-aldosterone system of the kidney. What happens basically? We all know that angiotensinogen is a peptide that is secreted by liver, and renin is an important enzyme which is secreted by kidney, and renin, by the name, it is the first component of the renin-angiotensin-aldosterone system. So basically, when there is decrease in renal perfusion, how can there be decrease in renal perfusion? Due to fall in BP. A fall in BP would cause a decrease in renal perfusion. This is a stimulus for the release of renin. Renin causes conversion of angiotensinogen into angiotensin 1. Angiotensin 1 in the endothelium of renal system and the surface of lungs, there is angiotensin convertase enzyme which converts angiotensin 1 into angiotensin 2. And angiotensin 2 is a powerful vasoconstrictor, and this powerful vasoconstrictor causes arteriolar vasoconstriction, and this is responsible for the BP regulation at minutes to hours. It single-handedly forms the intermediate wing of BP regulation at minutes to hours. Additionally, there can be ADH secretion. ADH is also called as vasopressin. And vasopressin also has a vasoconstrictory effect. And this vasoconstrictory effect again forms the intermediate arm which regulates BP at minutes to hours. The extended form of renin-angiotensin-aldosterone system, which causes an elevation of sympathetic activity, tubular sodium chloride reabsorption, stimulation of adrenal cortex to release aldosterone. These all processes actually form the long-term regulation of BP by the chronic arm of renin-angiotensin-aldosterone system. There are two additional mechanisms which we need to study. One of them is the capillary fluid shift, and other is a stress relaxation response. So capillary fluid shift, it occurs due to the action of Starling forces. Basically, as you can see in this image, this is the image of a capillary which is guided by a precapillary sphincter. So in a capillary, when there is a reduction of BP, when there is low BP is indicated by decreased fluid volume inside the capillary. And when there is decreased fluid volume inside the capillaries, it causes an increase in oncotic pressure. This oncotic pressure, it stimulates or it draws the water. It draws fluid from tissue space into vessel, and hence by this process it actually maintains BP. The opposite happens when there is increased fluid. The increased fluid actually causes a reduction in oncotic pressure and an elevation of hydrostatic pressure, which causes the release of fluid into the tissue space. This is nothing but the mechanism through Starling forces in which the fluid moves into or out of the vessels depending upon the hydrostatic pressure. Then the other mechanism is stress relaxation response. Stress relaxation response, it is mediated by the shear stress of blood flow that occurs on the vascular smooth muscles of blood vessels. How it happens? It actually happens as there is shear stress. The shear stress causes the activation of endothelial nitric oxide synthase. Endothelial nitric oxide synthase is responsible for production of nitric oxide. Nitric oxide is the chief compound that causes the relaxation of smooth muscles. And this relaxation of smooth muscles, it causes decrease of total peripheral vascular resistance and causes decrease in BP. So all of these mechanisms summarizes the intermediate regulation of BP as well. So this was intermediate regulation of BP acting through renin-angiotensin-aldosterone system, ADH, capillary fluid shift and stress relaxation response. Now let us move on to the chronic arm of renin-angiotensin-aldosterone system that causes the long-term regulation of BP. This process is called as pressure diuresis and pressure natriuresis. So it is again it is the similar process that we have discussed above. As we have seen that angiotensin 2 is responsible for tubular sodium chloride reabsorption, water retention, increased sympathetic activity and aldosterone secretion. The same process is happening when there is elevation of blood pressure. It causes increase of renal blood flow. It causes increase of renal blood flow, which causes increase in GFR, which again causes increase in urine flow. And this increase in urine flow is nothing but pressure diuresis. Increase in VP, it increases the peritubular hydraulic pressure, and hence there is increased blood flow to the kidney, which causes a reduction in renin release. A reduction in renal release inhibits the RAS system. Inhibition of RAS system, it decreases sodium reabsorption in the collecting duct, and that causes pressure natriuresis. So natriuresis and diuresis that are stimulated by pressure or basically blood pressure. It is the long-term regulation of BP by kidneys. This graph that you are seeing right over here, it is called as the renal function curve. It shows basically that at constant water and salt intake, which is indicated by this line, our body has an equilibrium point called as A. And basically as the salt and water intake increases, the equilibrium point shifted upwards, and that is indicated by elevation of BP. So basically when the kidneys become faulty and kidneys are unable to excrete salt and water, as in case of acute kidney injury or acute renal failure, there is hypertension associated because of inability to excrete salt and water. Now this long-term regulation of BP by kidney is one of the most important regulation because it has the highest efficacy of regulation. As you can see that even when there is elevation of arterial pressure by just 2 times, the urine output can become more than 10 times for the regulation of BP. So this actually shows that how much highly efficacious the renal regulatory system of BP is. So this basically summarizes the long-term regulation. Additionally, there is one more mechanism, that is atrial natriuretic peptide and brain natriuretic peptide, which are antagonistic mechanisms of renin-angiotensin-aldosterone system. They are increased in response to atrial and ventricular stretching, and they promote natriuresis. So this last image is basically showing all different mechanisms of pressure regulation in our human body. You can see 3 different responses: CNS ischemic response, baroreceptor and chemoreceptors, which are activated within seconds. Then there is stress relaxation and capillary fluid shift, which is actually activated in minutes. Then there is activation of renal systems, especially the early phase of renin-angiotensin-aldosterone system mediated by angiotensin 2 and antidiuretic hormone, which is activated within hours. The chronic phase of renin-angiotensin-aldosterone system actually is activated within days, and it is mediated by aldosterone, and it is counteracted by ANP and BNP. So this summarizes basically the regulation of BP. So to summarize, let us again look at what were the three major arms. So there was short-term regulation, intermediate term regulation, and long-term regulation. Short-term regulation is mediated by baroreceptor reflex, chemoreceptor reflex, and CNS ischemic response. Intermediate term regulation is mediated by kidneys through renin-angiotensin-aldosterone system, ADH, additionally by capillary fluid shift and stress relaxation response. And long-term regulation is again under the control of kidney through pressure natriuresis and diuresis, and the chronic effect or long-term arm of renin-angiotensin-aldosterone system, and this process is counteracted by atrial natriuretic peptide and brain.